Welcome everybody. This is the first ever IHMRI webinar series that we've done. Obviously, due to COVID, like a lot of organisations, we've had to take our events online. But we're very happy to have you here today. So I'm here with Professor Marie Branson. So welcome, Marie. Thank you so much for taking time to be a part of this webinar today. I'd just like to tell you a little bit about IHMRI. For those of you who don't know, IHMRI, the Illawarra Health and Medical Research Institute, we are a not-for-profit charity, and our goal is to support health and medical research right here in the Illawarra. We cover all sorts of different research into all different types of diseases, from cancer to mental health to diabetes to dementia, just to name a few. And in case you're not aware, the Illawarra Shoalhaven area is actually a really special region because of its diversity, and it proves, and it's been proved to show a really good snapshot of Australia right here, just in our little region. So it's a really great area for the research to be happening. And before we get started, I just want to say a very quick but large thank you to all of our staff, researchers and students at IHMRI. I know during COVID there have been lots of different changes, and our staff and researchers have had to really adapt to all different ways of working, and I know that's taken an incredible effort to, for example, get teaching online and to allow research to continue. So I just want to say a big thank you to everybody involved for making that happen. And of course, a big thank you to our community supporters, those organisations and individuals who help IHMRI continue doing what we're doing. We're very grateful, so thank you. So just a little bit of housekeeping. If you have any comments, you can just pop those in the webinar chat. And if you have any questions that you'd like to ask Marie, please pop those in the Q&A box. I'll be collecting all of those questions, and then I will field those to Marie at the end of her presentation. So let me introduce you to Marie. Marie is an IHMRI Senior Fellow whose research focuses on cancer metastasis, biomarkers and therapeutics, the plasminogen activation system in tumour invasion, and metastasis and targeted drug development against biomarkers of cancer and inflammation. So thank you so much, Marie. Over to you.
Okay, I need to share my screen.
Yep.
All good?
Yep, wonderful.
Okay, well, thanks, Kate, and thanks, IHMRI, for giving me this opportunity to talk about myself today. My research group focuses on cancer research, and particularly on the process of cancer's spread and invasion into other distant organs from a primary tumour known as metastasis. And we're looking at what drives it, and if we can find markers, or what we call biomarkers, that help those tumours metastasize, can we develop drugs to target that? But before I go into a particular project, the skin cancer project that we're working on, I thought you might like to know a little bit about me and how I got to be where I am now in a super whirlwind tour of my life. Okay, move on. So I'm actually a born and bred Wollongong girl. So here I am at Fairy Meadow Demonstration School, and can you guess who's me in the lineup? There I am. So yeah, I grew up in Wollongong, and that was a very happy childhood, to Italian migrants. I then couldn't wait to get away from Wollongong in those days. Now I think Wollongong's a fantastic place. And I did my honours undergraduate degree and did an honours in biochemistry at the University of New South Wales. There I focused on fatty acid metabolism as one of the various metabolic pathways. But then, as happens in science, I changed fields completely to do my PhD. I did that at the University of Sydney, and there I did a project which looked at the normal pigment cells in our skin, known as melanocytes, which can sometimes manifest themselves as freckles, and how they are different to melanoma, which is the malignant form of the melanocytic cells. So I did that for my PhD, and then I moved on again from this field. I travelled around after my PhD for a year, and then I found a research fellowship or postdoctoral fellowship at the National Institutes of Health in the US, in Washington, DC. I was there for 2.5 years or so. That was a great place to live, and I really enjoyed being there. And it's one of the perks of being a scientist. It gives you a ticket to travel and work all over the world. But there I actually got involved in developmental biology, looking at so-called homeobox genes, the genes that drive development. And I was using chicken embryos, there's a picture of one, as a model, and I was looking at the genes that control the wing or forelimb and the leg development. So that was a real move away from what I had done for my PhD, but this really got me into molecular biology techniques, which was really just taking off then, and that was a really good learning experience for me. Then I came back to Wollongong. There was a position going at the University of Wollongong where I was a research fellow there for a few years. And then there was an opening for a research and teaching academic position, which I was lucky enough to get. And so that's where I've been since 1999. But during that time, I did some stints as sabbatical or study leave in various countries over the years, and for 6 months or so. And that has also been great because it's allowed me to really cement collaborations with international research teams, and also it's just exposing you to different ways and culture. So at the University of Wollongong, I have a lab, and I do various types of cancer research. But in this presentation today, I'm going to focus on the skin cancer research we're doing. So I did start off my research career in skin cancer, and here I am back again doing it. But I worked with melanoma in my PhD, and that's, most of you would think of skin cancer and melanoma as the same thing, and they are. Melanoma is a skin cancer, but the skin cancer that I'm working on now is one of the most common skin cancers. Melanomas are not that common, thankfully. And there's two types of those more common skin cancers: is basal cell carcinomas and cutaneous squamous cell carcinomas, or CSCCs. These, as I said, are the most common of all cancers. And I'm going to focus on cutaneous squamous cell carcinoma. It actually causes quite a few deaths from skin cancer in Australia as a non-melanoma skin cancer. And you can see that's getting up there towards melanomas. So it is actually causes more deaths worldwide than many other cancers. So cutaneous squamous cell carcinoma, or CSCC, it's most commonly found in the head and neck. There's a rising incidence of CSCC worldwide, partly due to increasing aging population, partly due to higher UV exposure, and this goes for people from the Northern Hemisphere, from cold climates, who then go and have their holidays in hot countries and overexpose themselves to sun, to getting a tan, and they are getting increasing incidence of this disease as well. And people who are immunosuppressed also have a higher risk of CSCC. Not surprisingly, CSCC is a disease of UV exposure. A significant proportion of cases present as high risk, and this is due to invasion of the upper layers of the skin, as shown here, into the underlying dermis or subcutaneous tissues. So it's normal skin plus UV is actually looking a little bit damaged because of the UV damage. And here is a skin cancer, a CSCC, and you can see that it looks very different to normal skin. And this tumour infiltrating down into the underlying tissues. Now, this project in the CSCC, it developed in my lab because I have to blame a local surgeon, Prof. Bruce Ashford, who deals with this disease all the time, the surgery on this disease. who had many questions about the biology of the disease because it wasn't that well understood. So he came to my lab and asked a few pertinent questions, and together we started working on it. And he actually did a PhD in this area, which he completed last year. And I learned a lot of things about this disease from him. Now, only a small proportion of this disease will develop metastases, will spread from the primary tumour. Now, that doesn't sound like a lot, but you've got to remember that there are hundreds of thousands of cases of CSCC a year in Australia alone. So 5% of that big number is quite a few people. And Bruce sees quite a few patients in his clinic. These normally, usually, from a head and neck region, will spread to a local lymph node, often around the neck region. And this significantly increases the risk of morbidity and also death. This just shows a CT scan with tumour in the parotid gland, which is in the neck region, and the lymph nodes in there. So it's one of the most common lethal cancers, metastatic cutaneous SCC. Bruce sees at least 80 cases per year in Wollongong alone. And unfortunately, as I said, that causes a lot of morbidity. So it requires quite big surgery to remove the involved lymph nodes and other regions of the head and neck. And sometimes, often, not often, it's followed up by radiotherapy, but there are no systemic or drug treatments that is common for this, commonly applied to this disease. So you can see this is quite disfiguring. The disease can be recurring and it can be lethal. It can go on to kill people. Now, the thing with metastatic CSCC is these predictors of high risk, like the tissue might be sent to a pathology lab and it's invaded into subcutaneous tissues. It's got other suggestions of this primary maybe has spread further, but these don't always get it right. So, for example, there's quite a few of these so-called high-risk tumours that don't metastasize or will never metastasize. And here's an example of a really large CSCC on the side of the head of this patient. It was large, it was thick, it was deep. But this patient's tumour never metastasized, and he underwent quite significant surgery to look for involved lymph nodes, because on suspicion of this being a high risk, that it had spread, but in fact it hadn't, and this patient could have been spared of that radical surgery to look for these for metastases that weren't there. So this is where Bruce came in, really saying, well, you know, I operate on these people, sometimes I don't need to operate them, sometimes I should be operating on them earlier. You know, the risk factors just don't really predict well enough whether or not we should be treating a patient more aggressively or not at all. So then we said, okay, well, we don't really know much about what drives metastasis in this condition. And if we could work that out, can we then use that information to predict which tumours or primary tumours will spread or not, and so which patients should get more attention to their disease. So Bruce's PhD project was based around looking at several of these metastatic tumours from lymph nodes, and he employed so-called whole genome sequencing, which basically looks at the DNA blueprint of the tumour cells. We compare that to normal cells and looking for mutations or damage to the DNA in the tumour cells. And this picture here is called a Circos plot, and it's just a snapshot of one patient's DNA blueprint from the tumour cells. And it's basically all the chromosomes. And what it shows as you go out from in, all the different types of alterations, mutations, rearrangements, damage to the DNA of that tumour cell. And this actually shows there's quite a bit of damage to that cell. Now, this project and the ability to do this whole genome sequencing, which was and still is reasonably expensive, wouldn't have been possible without a generous donation from the local charity group, the Illawarra Cancer Carers, which really kicked off this project. And we thank them very much for that support and getting this project off the ground. Then once you have the DNA looking at mutations, that's all well and good, but you also need to know what the result of those mutations are to the functioning of the cell. And to do that, we look at an intermediate molecule between DNA and the proteins in cells that affect how the cell works, called RNA. This allows you to map the impact of the DNA mutations. And so this is also being done. And we integrate or combine the mutation data with the RNA data to see whether the mutation actually has an impact or not on the functioning of the cell. And then again, the Illawarra Cancer Carers stepped in to help with this type of work with a $130,000 donation, which allowed us to buy this high throughput RNA analysis machine called a nanostring. And so that has helped us to analyse the RNA of multiple tumours so far, and we get these kind of heat maps that show differences between metastatic and non-metastatic cancer. So we're trying to come up with some sort of signature that predicts whether a tumour will metastasize. And, you know, because we had this money and we could do this work and we got very nice preliminary data, this allowed us to apply for Commonwealth grants, like the IHMRI grant, which took us a couple of years to succeed, but we finally this year got a $600,000 grant from them, which is allowing us to analyse up to about 100 patients. So Bruce did 15 in detail, and this has been published now, but to really build on the statistical significance of this and to go across the variation that exists, we need to look at more patient samples, and that is going to help us look at more patients. And I have to say that this work is a inter-institutional collaboration between these institutions, including, and also a colleague in Singapore, National Cancer Institute in Singapore. So definitely it's work that it goes across multiple institutions and not just done by our group. And that's important in being able to do this sort of scale of work. So just to summarise, so the CSCC research that we're doing here at IHMRI in collaboration with people across the country and overseas is looking to understand the biology of metastasis in CSCC. So we're hoping to understand how it works. And from that, hopefully, and we are coming up with evidence of potential biomarkers that help identify CSCCs that are at risk of spreading. And by doing that, those biomarkers could turn out also to be very good targets for new therapeutics. And just to show an example here, so this is a lymph node, and this is a lymphatic, this around here, which is feeding into the lymph node. And what you can see here, this clump of cells is actually captured in situ, is a tumour embolus or group of tumour cells that are in the lymph node, that are making their way, in the lymphatic, which are making its way to the lymph node, where this is metastatic site, where it can spread from there. And it's stained brown for a biomarker that we have identified as being upregulated or higher in tumours that are metastatic. So I will stop here for questions. These are just the collaborators, and I'd like to thank them, acknowledge them for their work in this project. So, Kate, hand it to you. So I'll stop sharing.
Great, thanks, Marie. Yep, if you could stop sharing your screen, that's great. Thank you very much. Well, gosh, thank you so much, Marie, for that presentation. We do have a few questions that are coming in, and I guess some of the questions are relating to, for example, the early symptoms of a squamous cell carcinoma. And I guess I just want to acknowledge that, Marie, although you work very closely with Bruce Ashford and other colleagues at the hospital, you're not a clinician, but is that something that you would be comfortable answering? Or
what are the symptoms?
What are the early symptoms? What should we look out for in a squamous cell carcinoma?
Yeah, they can. They look sort of scaly, and they can be a little bit. What's the word? A bit like an open sore, but they can vary, you know. Yeah. And they can be confused with a keratosis, and sometimes confused with a BCC. So look, yeah, I'm not a clinician. If you have something that doesn't look right on your skin, always get it checked out. Yeah. Even if you think, oh, it's nothing, it's definitely. And I do. I actually often get what I think might be something just, you know, burnt off with the CO2, the liquid. What's it called? The dry ice?
Liquid nitrogen.
Liquid nitrogen. Thank you. God, just had a brain freeze there. So just in the doctor's surgery, I don't like this. Just freeze it off. But if it doesn't look too good, they'll obviously do a little biopsy and send it off to the pathologist.
Yeah. So the questions that have come through relating to examining anything that doesn't quite look right, I won't ask those questions. I think the message is just to go and see your doctor and get those checked out.
Mm-hmm.
Scott Griffith would like to know, thanks for your question, Scott, how quickly does CSCCs grow, and is metastasis likely to occur early in the presentation?
That's exactly what we're trying to answer. That's, as I showed you with the fellow that had the 10 centimetre thick. So he presented with that, you know, pretty late.
Yeah.
You know, you want to go a lot earlier than that. and it looked for all intents and purposes like it had spread, but it hadn't. Whereas someone might have something a lot less nasty looking, and it might already be spreading. And we don't really know. We don't really have the answers to that yet. That's what we're trying to work out. Yeah.
Okay, so some other questions here. The reason we're doing the webinar is because of COVID-19 and not doing a face-to-face. So we just want to know, has COVID-19 impacted your research at all?
Not really, I'd have to say. We've been able to keep the lab going. Mm-hmm. There was a bit of touch and go at the beginning, you know, do we shut down the labs, do we not shut down the labs? But in fact, the labs are really clean, safe spaces. And so we felt that as long as we can maintain as much social distancing as possible in the labs, IHMRI's been really good at setting up those processes, so not crowding people in at the same time. So it has slowed things down a little bit, because sometimes you can't go into a lab because there's already too many people in there. Mm-hmm. You know, but otherwise, no. Noticing a bit now that it's taking a little longer to order in some reagents. Some of these things, or many of these things, come from overseas.
Yeah.
So that can take a little bit of
supply slow down
a little bit. Yeah.
Yeah. Okay. Well, that's great to hear that your research has been able to continue, and people are just using common sense to make sure that the social distance in the lab. So you're involved in a lot of different projects, Marie. Why was it this particular project that you're more drawn to, or that that is your current focus?
Oh, well, I focus on I got quite a few projects going. I thought this one would be interesting to talk to in a community sense, because I guess, you know, this country, we're rife. The skin cancers are rife in this country, and I thought it might generate more interest that way. But this project really, I really didn't know that much about CSCCs until Bruce came along, and I was quite blown away by how prevalent it is and how little we know about it. And I thought also this is a fantastic opportunity to work with a clinician and other clinicians from Sydney who are really research focused and oriented, to work with clinicians very closely on a disease, rather than me just working away in the lab. Yeah. In an academic sense, if you know what I mean. It has a real clinical, what's the term? it's really clinically relevant. Yeah. It's quite exciting to work alongside clinicians who see it every day, who work with this all the time, reminding you why it's important to do.
Yeah. It's wonderful to hear this collaboration as well. As I said earlier at the beginning of the presentation, IHMRI was created to help that transition from academic research to the clinical space.
Yeah.
For the outcome of better patient outcomes in the long run. So it's wonderful to see this in action.
Yeah.
What are some of the major challenges that you have in your research?
Generally speaking?
Yeah.
Okay, I reckon, I say this quite a bit, I think a major challenge is keeping, just, the old adage, you can't get good help. I've got good help. I've got great people in the lab, but they're rare. And funding is always short term. If you're lucky, you've got 3 years of funding to keep somebody around. And they become highly skilled in the methods that you use in that project. But if you can't get continuous funding, you have to let them go. So you lose that skill base. So there isn't really, in Australia, there isn't a career that is research assistant that is continuously funded. It's people like me getting grants to try and get your research assistant and try to keep them and not lose them. And we rely also on students, on PhD students who are good and they're learning. But once they're done, they want to move on to other things. That's fine, of course. So I think the biggest challenge is having the skill base there on the ground and not losing it and reinventing the wheel when you get somebody new in the lab. That slows things down a lot.
I guess it's great to see the support that you've had from the community, from the Illawarra cancer carers and others, just to help you with your research and ensure where possible that can continue.
That was, we wouldn't have, I can say almost 100% we wouldn't have gotten that big government grant if we hadn't had that injection of funding from the cancer carers to allow us to get the information and data to say, we have got the capacity to do this, and we've got the data to show that it's worth pursuing a larger scale.
That's it. I've got a question come in here from the comment box. I'm not sure if you can see that one. Someone was wondering if you've had the chance to study a wide range of ages with SCC.
Look, most of the patients that we get are older people, and these are a handful of younger patients that come in, and that's very interesting to the clinicians and to us from a genetic point of view. Are they just unlucky, or is there some predisposition that allow them? Because as you may or may not know, cancer tends to be a disease of old age. Over time, you accumulate all sorts of sun damage, all sorts of air pollution, whatever, and that causes more and more mutations, and the body gets less able to fix those. And so with time, when you get to an age, your risk of cancer increases. In younger people, it's quite rare. And that may be for various reasons. It could be underlying genetic reasons that we're looking at.
So it's good to have those age differences so that you can have a look and dig a bit deeper into that question. So, Marie, thank you so much for telling us a little bit about yourself at the beginning of your presentation. It was great to see what's led to you, or how you've reached where you are now. If you hadn't become a researcher, what do you think you'd be doing?
Yeah, that's a really good question. I was quite interested in architecture. I contemplated doing that for my degree, and I don't know why I didn't. I'm kind of glad I didn't, actually. I think it's quite even more of a cutthroat industry. And I'm really interested in food. we all are. I wouldn't be running a restaurant or cafe. That's even more out there. But yeah, maybe doing something related to food, growing food, or prepping food or something like that, you know, that kind of thing, because I really do enjoy cooking and getting more into gardening.
What's your signature dish, Marie?
Oh.
Do you have a go-to?
Oh, I do pretty good seafood.
There you go.
Yeah, with pasta.
What is best spot to be living in? Wollongong, you've got fresh fish on your doorstep, and we're very glad that you did stick with research and that you're now working towards solving some of these questions that really research questions that really impact us as a community. So we're just about out of time. Thank you so much for putting that presentation together and answering our questions. We really appreciate it.
Thank you, and thanks everyone for listening.
So before everyone heads off, I'm just going to pop up, share my screen with you. Oops, next one. Just to say a big thank you to not just Marie, but also to our community, all you who've dialed in today. We really appreciate your engagement and your support. Thank you so much to everyone who asked some questions. It is Science Week this week, so we've got another presentation tomorrow by Dr. Monique Francois, who will be talking about cardiovascular health and whether a certain type of diet combined with exercise can help to reduce your risk of cardiovascular diseases. So if you haven't already dialed into that, registered for that, you can do so the same way that you registered for this on eventbrite.com.au, sorry. And also keep an eye on our social media channels this week. We've got a lab group, a different lab group each day taking over our social media. So there'll be lots of exciting activities and content on our social media. So that's facebook.com/IHMRI and Twitter is @IHMRI. And as I said, we are a not-for-profit. We do rely on the generosity of the community. So if you would like to make a donation, we would be very, very grateful. Go to our website, ihmri.org.au, and then click on support us, or just go straight to make a donation. So once again, thank you so much to everybody for being involved, and to all of our supporters. We really do appreciate your generosity and your support. So I will see you tomorrow. Thanks again. Bye.